And now let's look at the second half of Unit 6. A genome sequence is the complete list of the nucleotides that make up all the chromosomes of an individual or a species. Within a species, the vast majority of nucleotides are identical, but sequencing multiple individuals is necessary to understand genetic diversity. When you begin to actually compare organisms, we find that there are some basic trends. The gene density is higher in bacteria and archaeans than it is in eukarya. The overall genome size is significantly larger in eukarya. Introns exist in eukarya and in some archaeans, and we see lots of these little differences here. Okay, so the basic trends we see are that archaeans and bacterians, the prokaryotes, are more similar to each other as far as genome size and structure, but archaeans are showing some of those eukaryotic traits like introns. Prior to knowing the full genome sequences, estimates of the number of human genes range between 50 and 140,000. As genome sequence quality and the methods for identifying protein coding genes improved, we realized that it was probably closer to 20,000. When we actually work in RNAs and the various other structures that don't code for proteins, it goes back up to about 46,000. The numbers vary. Okay, but when you actually look at these structures, we find that there are some general things in common. You have a variety of traits and structures that exist, but protein coding sequences, those exon structures, only account for a very small fraction of the genome, about 1.5%, and the rest is associated with non-coding RNA genes, regulatory DNA sequences, long interspersed nuclear elements, short interspersed nuclear elements, introns, and all sorts of other structures. Okay. Overall, the lines, for example, are majority inactive, but they contribute to the instability and the evolution of the genome, and it's tightly regulated by DNA methylation and things like Pee-wee interacting RNA. The ALU elements, which are the majority of the signs for primates, play a role in evolution as they are highly conserved. Repetitive DNA that is not transposable is about 14%, with some of those being the simple tandem repeats, which are widely used for DNA profiling and kinship analysis and forensic identification. And the simple tandem repeats are also going to include things like telomeres. The large segment duplications, it means you only have a couple of copies, but for example, the Y chromosome is made mostly of large segment duplications. You have unique non-coding DNA like gene fragments and pseudogenes. These are related to known genes, but they've lost their protein coding ability or are no longer expressed. Regulatory sequences are going to be things like promoters, enhancers, control elements. So we see lots of different things going on with this structure. When you really break it down, about 8% of the human genome is old virus sequences. It varies, and the structure is going on is going to be different from organism to organism, but the basic components are the same. Transposable elements are sequences of DNA with a defined structure that are able to change their location in their genome. These are categorized either as class 1 transposable elements, which are replicated by a copy and paste mechanism, or class 2, which can be excised and removed from the genome and inserted at a new location. The movement of these elements is a driving force of genome evolution because their insertion can disrupt gene functions. Homologous recombination between these elements can produce duplications, can shuffle exons, move regulatory sequences to new locations, all sorts of things. We also have retrotransposons, which are transcribed into RNA and then duplicated to another site. These can be divided into long and non-long ones. I know the naming is not great, but this is also showing us that reverse transcriptase capability is essential to eukaryotes and not just viruses. When you look at the actual regulation process for both eukaryotes and prokaryotes, the first one we have to acknowledge is metabolic pathway regulation. This is going to be true for all organisms, prokaryotic and eukaryotic. And if you are having difficulties with this, you can take a step back to the metabolism unit. But efficiency is a major driver of natural selection, and so negative feedback is going to be a homeostatic mechanism that is involved in all organisms, regardless of nucleus presence. The most common example of regulation in prokaryotes is an operon. The operon structure is prokaryotic only, but bacteria like E. coli, they need amino acids to survive because, like us, they need to build proteins. One of these is tryptophan. If tryptophan is available in the environment, E. coli will take it up and use it. However, E. coli can also make their own tryptophan using enzymes that are encoded by 5 genes that you can see here. This example is a tryptophan operon. This is a repressible operon. So if tryptophan is present, if the bacteria is able to uptake tryptophan from its environment, it won't make it. But if tryptophan is not present, it can make it as needed. So the repressor in its normal state is on, but it can be turned off. So we can prevent the building of tryptophan if needed. And that's how this negative gene regulation works with the repressible TRP operon. The repressible operons are typically biosynthetic, building molecules up. You also have inducible operons. These are typically digestive or breaking things down. We'll find that we're looking specifically at the lac operon. Lac is produces the enzymes needed for digesting lactose, and the bacteria is not going to produce lactose if it doesn't need to. So it's inducible. When lactose is present, it will bind to the repressor, inactivating it and allow the production of the lactase enzymes. So overall, we find that there is a major difference here in the negative gene regulation. Both of them are negative, they prevent something, but repressible operons focus on biosynthetic pathways, and inducible operons focus on degradative or digestive pathways. We also find that there is positive gene regulation. This is going to cause an increase, not a decrease. So in the example of lactose and lactase, it's really a secondary sugar. Glucose is going to give energy much more efficiently. So if glucose is present, it's going to be digested preferentially. If both are present, then lactose will be digested as sort of available by chance. If glucose is rare, but lactose is high, then the secondary messengers like CAMP that would be involved in the glucose breakdown are going to upregulate using the CRP binding site, the CRP protein. This is going to cause an upregulation. This is going to cause the RNA polymerase to bind more rather than less, making it a positive gene regulation. Eukaryotic gene regulation is significantly more complicated because there is a separation with the nucleus and the cytoplasm. So we don't just have this operon-like structure. Well, we do have things that are like an operon, but we don't have operons because operons can only exist when there is no nuclear separation, because transcription and translation have to occur in the same space. We find that there are many different levels of control in a eukaryote, and you can see each of those sort of indicated in the picture here. But each level is separated, and each level provides a different point of control, drastically increasing the efficiency of eukaryotic regulation. The first level of control is chromatin modification. By binding acetyl groups, you push the nucleosomes further away from each other by altering the charge connection. So this is going to open up enzymes and access of enzymes to gene components and increase the copying of whatever components are there. We can also deacetylate, which will reverse this process and bind them closer together. The more condensed, the less access there is for the enzymes to read the process and to produce the mRNA. The secondary level of control is going to be that of transcription. We find that there are control elements. Enhancers are the distal ones that are far upstream, and then we have the proximal control elements that are nearby. And then at the end, you also have termination regions. So all of these signals are going to serve as binding sites that are going to alter the binding of transcription factors that are going to lead to the production of very specific mRNA sequences and producing the proteins that you need in the long run. So eukaryotic transcript production is controlled by what binds to these active control elements. Those control elements, it could be an activator. We could have DNA binding proteins that cause the general curvature of the structure, but those distal control elements are brought in line with the proximal control elements. That creates a binding site for the general transcription factors that are needed for all things, and the specific transcription factors, the mediator proteins that are needed for that specific product. The binding of all of these components creates an even sort of an upregulated binding site for polymerase II and encouraging that activation. So this is kind of like that positive control element. But everything is controlled with these enhancers and these activators, and which activators and which enhancers are triggered will control what is exon, what is not, which exons are being bound. All of these processes are controlled. So a liver cell and a Lin cell, for example, could be producing the exact same DNA segment, but produce very, very different things due to the transcription factors and the enhancers and the activators that are binding. All of this working together to increase the complexity and to increase the efficiency of our genome. So we don't need an incredibly high genetic density like a prokaryote does because we have so many levels of control. All of that works together to control the RNA processing, the introns that are present, and alters which exons remain in the final product. So intron and exon, that is determined by those small nuclear RNAs, and what the small nuclear RNAs are binding to is determined by the transcription factors that are attached. So I can make very different products just by producing a different pattern of exons. It can even get more complicated when I try to combine this with the idea of transposons actually moving the exons around. So in the general process, all we can do is not make something an exon. But in the actual complexity of life, it gets way more complicated. When you look at the non-coding RNA, that's our next real level of control. Once we've produced the RNA, I can either let it go or stop it. And I find that this is where those small interfering RNA and the microRNAs are going to be involved. These are both smaller RNA molecules that mediate gene regulation using the same mechanism. Both are processed by a dicer enzyme and loaded into the RISC, or the RNA-induced silencing complex. And by binding with the mRNA, it degrades the mRNA or aborts the gene expression. So basically, this is putting a pause or a destroy on an mRNA and regulate what it's producing. So if I have an mRNA that's not doing the proper thing, or if it's producing something that I no longer need, then I can interfere with it using RNA. Just like in transcription, translation has initiation factors as well. We talked about the binding of the mRNA to the small subunit of the ribosome. It turns out that there are other proteins and molecules that aid and encourage that process to occur and to produce very specific things. So transcription factors encourage the production of transcript products, or mRNA, and translation factors control the production of proteins. Those proteins that come out will then need to be folded properly. That word might be where a chaperonin will come in to aid in the folding process. And if the proteins cannot fold properly, if they are misfolded, they will be labeled with ubiquitin and will either be carried to a lysosome to be digested or taken to a proteasome to be destroyed. We cannot have misfolded proteins. That is where we get many different diseases, as well as pathogenic proteins like in the prion diseases. Overall, we see that mutations can alter what we produce and lead to genetic disorders. So genetic disorders are much smaller scale. You're looking at nucleotides changing, not whole segments of chromosomes. We do need to realize that not all mutations are a bad thing. Sometimes mutations are just neutral, sometimes they're beneficial. But of course, we're looking mostly at negative examples or genetic disorders. Sickle cell is a great example of this because it's a so-called point mutation. You have one single nucleotide change that leads to these wide variety of negative physiological effects. So mutations don't have to be big to have a major effects, but those effects don't have to be negative. The most common type of gene mutation is a substitution mutation. You're taking one amino acid and you're replacing it with another. That can have three results. It could be a silent mutation where because of wobble, you're replacing with the same amino acid. So glycine becomes glycine. There is no negative effect. There is no effect at all. It's silent. You could change glycine to serine. That's going to be a missense mutation because you've altered the amino acid. Or it could be a nonsense mutation where you change to a stop codon. But a substitution mutation is a point mutation with a single nucleotide being substituted, leading to negative or neutral effects. We could also have insertions or deletions. These are the adding or the removing of bases. This is going to lead to a frame shift mutation. So because you're altering the codon, everything downstream could be altered. So a deletion of one could be minimized due to wobble, A deletion of 2 is going to be our worst case scenario because it's going to shift everything downstream, and wobble can only help so much. And a deletion of 3 is going to be kind of your best case scenario because all you're going to do is lose 1 amino acid. So it could have an effect, it could not. It depends upon where it is. A mutation like this at, say, an active site is going to be significantly more impactful than somewhere in just a transmembrane structure that has minimal effect. Overall, we see that mutations contribute to genome evolution. We have whole segments of our genome we talked about that contribute to genomic instability and variation and evolution. Traits that are highly conserved must be essential to life and must be important for evolutionary change. But just because you have these lines of descent from a common ancestor doesn't mean that the chromosomes will be the same. Humans and chimps have different numbers of chromosomes because at some point in our ancestry we had a fusion event that chimps did not have, or vice versa. I honestly don't remember. And then human and mice, we can find that divergence and duplication and the various mutation events have caused the genes on the human chromosome to be separated over multiple chromosomes in the mouse. So evolution is connected, and we can use biotechnology and biochemistry to find out those connections and contribute to the broader understanding of evolution. So duplication and divergence is one way that these things can move around. Because crossing over happens, because you have incorrect pairing of homologs, you can cross these things between different chromosomes. It can happen. It doesn't have to be correct pairing. And that can lead to changes. That can also lead to examples like in hemoglobin, where you have these broader gene families. Multiple duplication and divergence events have led to the production of multiple genes that work together to produce one thing, and that has slowly led to increased efficiency and change and evolutionary benefit. Evolution is a change in these things, but we can also see that it's tied in with conservation. Many different processes, duplication and exon shuffling, can create changes in the organism, and that can lead to massive change through life, especially if you're looking at those master regulatory genes. Exon shuffling is a key mechanism here because 2 or more exons can be brought together, they can be duplicated, you can make all sorts of events. The Hox genes are master regulatory genes that control the distribution of the up-down gradient of the body, and duplications of Hox genes are one of the major changes, the major sources of evolutionary difference between living organisms. Biotech is going to be our last major section of Unit 6. We've done biotechnology from its most basic level through animal husbandry for millennia, but we're looking directly now at things like artificial selection, so that's controlled breeding, genetic modification, GMO, genetic engineering, which means you're taking specific genes from one organism and placing them into another. All of these things are possible because of the universality of the genetic code. So the so-called farm animals, PHARM, we've taken the gene for a human gene product and we've put it into the milk production genes of the goat so we can extract medicines from it. Okay, in the vegetables on the right, we've increased the amount of vitamins to decrease child malnutrition. So some of these things can be very beneficial, but we have to do a lot of study to find out what is actually going to be the most benefit. We realized fairly early on if we wanted to be successful at genetic engineering, what we first had to be able to do was to sequence the DNA. You have to be able to determine the nucleic acid sequence in order to really make changes, because if you don't know the sequence, you cannot alter that process. Now, the early methods include things like the shotgun method and the chain termination method. The shotgun method, you make lots of small fragments that you code individually, and then you use computers to overlap those codes. It's very, very fast, but it can be inaccurate, especially in areas of high repetition, like a centromere or a telomere. The chain termination sequence allows the production of DNA molecules that are each progressively one base longer than the last, using a dideoxynucleotide. That means that you're going to have these chains that can be ordered by length, and then you can follow the angle down to get the sequence. This process is going to be very, very slow, but very, very accurate. In reality, we use both of these things. We use the shotgun method to do the sections that are non-repetitive, and we use the chain termination sections to do the repetitive ones and combine those into a more accurate answer. Gel electrophoresis, so you can actually use gel electrophoresis to separate DNA fragments by size because DNA is negatively charged and moves towards the positive pole in an agarose gel. The smaller fragments will move faster, and you can use this to compare gene sequences. So when you go back to things like the STRs or the short tandem repeats, the fragments that this gel is broken into due to restriction enzymes will be different from person to person. And this can be very useful in things like paternity testing or forensic testing. DNA fingerprinting, for example, looks at the RFLPs, or the restriction fragment length polymorphisms, and looking at the short tandem repeats. So because different people are going to have different numbers of these STRs, the restriction fragments are going to be different sizes, and we can use a comparison of those to fingerprint for identification in criminal cases as well as paternity cases and a wide variety of use. The first successful case of using this DNA evidence was the 1987 rape case convicting Tommy Lee Andrews and releasing Earl Washington, showing that a great deal of information can be extracted from DNA samples. Restriction enzymes are used to cut DNA at precise locations. Because DNA is a very long molecule, it needs to be cut into smaller pieces to facilitate study and work. Restriction enzymes cut DNA at particular nucleotide sequences. These sequences are so-called restriction sites, and they're typically palindromic to the complementary strand. These restriction enzymes evolved in bacteria to cut up foreign DNA, and it protected them against viruses and other bacterias. So this evolved as a bacterial immune response, and now we take advantage of it. But we can see that palindromic relationship here with that GAA-TTC. You go to the complementary strand and reverse it to GAA-TTC. So if you see that kind of repetition, you're likely looking at a restriction site which can be taken advantage of. And then when you combine DNA ligase, you can glue the DNA back together and hopefully be able to put new strands in, creating a recombinant plasmid. When you make a recombinant plasmid, it can be very hard to see if you were successful. So we often use screening tools like reporter genes that you attach to the regulatory sequences to know for sure whether the reaction worked. This could be some form of antibiotic resistance. It could be some sort of fluorescence. But the reporter genes are used as an indication of whether a certain gene has been taken up by or expressed in the cell. Overall, this is going to know if you were successful or not, and is an essential component to any recombination product. When you're actually looking at DNA sequences that have been extracted, we find that if you're looking at eukaryotic sequences, introns can be very problematic. So complementary DNA is DNA synthesized from a single-stranded RNA, mRNA or microRNA or whatever, and catalyzed using reverse transcriptase. So that reverse transcriptase will allow you to produce a DNA strand that can then be used for recombinant plasmid production or long-term storage, what have you, without the intron mistakes. So this allows for us to produce things like the spike protein of the COVID vaccine because we're able to get the exact gene sequence of the protein, not of the broader DNA source. So we're not wasting a lot of DNA, we're getting the exact product that we need, and it can be very useful, increasing the efficiency. Once you have a DNA sequence that is usable, you can clone it in something like a bacteria, but that takes time and a lot of steps. So the PCR process, or the polymerase chain reaction, is a method of cloning DNA without all that process. The key ingredient here is Taq polymerase, primers, template DNA, and nucleotides. The ingredients are assembled in a tube along with cofactors needed by the enzyme and are put through repeated cycles of heating and cooling. The heating breaks the hydrogen bond through denaturation. The annealing or the cooling allows the primers to bind so the complementary sequences can be produced. And then the extension step, to step 3, is going to be raising the temperature again so that the Taq polymerase can extend the primers. Why do we use Taq polymerase? Well, we're doing this at things like 72 degrees Celsius. So if we used a human polymerase, it would never work. But the Taq polymerase coming from the Thermus aquaticus archaean functions at this higher temperature and allows us to clone without the need of doing it inside a cell, which can be very efficient. Once you actually have the gene products, you need to analyze it. Reporter genes on their own are very useful, but if you need to analyze many different things at once, a DNA microarray is going to be your most common tool. This is a collection of microscopic DNA spots attached to a solid surface, and you measure the expression levels of a large number of genes simultaneously. Each DNA spot contains a small amount of DNA sequence known as probes. This could be a short section of gene, DNA element that's used to hybridize, like a cDNA or whatever. And then the probe target hybridization looks at fluorescence or chemical levels or light or what have you to determine the relative abundance. The core principle here is that the property of complementary nucleic acid sequences is essential. So a high number of complementary base pairs means a tighter bonding between the two strands. So after you wash off the non-specific bonding sequences, only strongly paired strands will hybridize and fluoresce. Gene cloning, of course, can be done in PCR, but if you don't have PCR technology, then you can do it inside a plasmid, which then goes into a bacteria and it is reproduced. We can't do this into the bacterial genome because it's very difficult to get a bacteria to accept outside DNA into its chromosome, but plasmids are significantly easier. So once we engineer the plasmid, we can allow the bacteria to grow, and then we can use that bacteria for a variety of things, like inserting pest resistance, eating oil, altering medication, all sorts of things can be used here. So the genetic engineers have to find specific genes that are of interest and useful, and then insert them correctly and ensure that it processes. But the technology is there, and it is fundamentally possible. If gene cloning is possible, we also need to talk about organismal cloning or reproductive cloning. This is the procedure of creating a new multicellular organism that is genetically identical to another. In essence, this form of cloning is asexual reproduction with fertilization or intergamete contact not taking place. This is very easy to do in plant species because they stay pluripotent throughout their entire life, their cells do, so we can clone those very simply. In a eukaryotic cell, we use somatic cell nuclear transfer to create animals that are genetically identical. This process involves the transfer of a nucleus from a donor adult cell, somatic cell, to an egg that has been enucleated. That is then hit with a little bit of UV energy or a little bit of electricity to begin the division process. If the egg divides normally, it's transferred to the uterus of a surrogate mother. They're not strictly identical, like you can see in the example below. CC, or carbon copy of the cat, was different from the mother because of X inactivation. Additionally, the mitochondria in the cytoplasm also contains DNA, so you will have differences in mitochondrial DNA depending on where the cell came from. This may have important implications for cross-species nuclear transfer, where there may be mitochondrial incompatibilities, so we're still having to do a lot of work with this. Dolly the sheep was the first mammal to have been successfully cloned this way from a 6-year-old biological mother. And it took 434 attempts. So this process is far from 100% control, but we are getting better and better at it every year.